These are notes on different types of gene expression, and it's really a summary to help you piece together everything you've learned this unit and to make sure you understand the different things we've talked about. So we've talked about gene regulation, which determines if genes are turned on or off. And you know that there are different proteins, activator and repressor proteins, that attach to the regulatory region of DNA that are based on environmental or chemical signals. So in this cell here, this is where lactase producing gene is activated. So this is the regulatory region, and this is the coding region, and this would be a piece of a chromosome. It's just one gene. Hopefully by now you know that chromosomes contain many, many genes. And it's activated. There's the activator, the little pentagon activator protein. In this cell over here, though, there is a repressor protein that attaches in front of the activator protein, and it blocks any signals. So really what's going on is that this little protein here helps this regulatory region send signals to the coding region that then allows it to be transcribed and translated. And then obviously that mRNA goes into the cytoplasm and the proteins are made. But here, this repressor protein blocks any signals coming from this activator protein, and therefore this region never gets transcribed. And if it's not transcribed, it can't be translated and it can't be made into a protein. And so that's how we could see how somebody could be lactose tolerant or intolerant based on these activators or repressors. But then we also have this other idea of functional and non-functional genes, and those describe the proteins made by the genes you have. So this is a different gene. This is the gene that makes the lactase repressor protein. So this protein here, and actually this one here, but we're going to focus on this triangle one here, is made by another gene. This is the gene that makes that protein. And you'll notice in both cases it is activated. So here's the activator protein for that gene. And so in both cases protein is being made. There's signals being sent for transcription and translation. But here, for whatever reason, at some point during like DNA replication, a G, here's this G, instead an A got substituted. It's just a mistake. It was an accident, no big deal. But the big deal, I mean it is a big deal, but the big deal is that then when this gene is transcribed and translated, instead of being this nice triangle protein, you get this sort of triangle with the top lopped off of it, right? So this is now a different shape, and the different shape now has a different function. What it can't do is it can't attach on here and repress this gene. And so because of that, because this can't attach on where it's supposed to, you get somebody who stays lactose tolerant, or we actually call that lactose or lactase persistent. The lactase gene persists after anywhere between age 5 and 15. Because we've talked about the fact that everyone is born lactose tolerant, which means everyone has a functional lactase producing gene, but at some point it gets turned off by a functional repressor. But if you have this mutation here, just that little change, then when that gene is transcribed and translated, you get a different amino acid for this set of bases, right? We know they come in sets of 3, so this is actually the methionine codon, which ends up being the start codon. And then whatever, and I don't know, but whatever amino acid GCC codes for, instead we have ACC, so that's going to be a different amino acid, so that's going to be a different protein shape, so it has a different function. We say different function like it has a function. Really it doesn't have a function, so it's just kind of hanging there, and it can't shut down your lactase producing gene, and so you stay lactase persistent. So now we can combine those two ideas. We can combine the gene version you have, so the functional versus non-functional, with the environmental stimulus of the repressors and activators. So we're going to look at the C. elegans that we looked at the start of the unit. And we looked at wild type C. elegans, and hopefully by now, based on the fruit fly eye, you know that wild type means that whatever trait you are looking at, those genes are functional. So it doesn't mean every single gene in the entire C. elegans is functional. It just means if we're talking about glycerolase repressors, if we say wild type, we mean the gene that codes for glycerolase repressor is functional. In a fly eye, if we say this is a wild type fly for eye color, we mean, there's like 8 genes in that pathway, and we mean all 8 are functional. There's also a mutant C. elegans. We were going to look at the mutant C. elegans this unit, but we just didn't have time. And that mutant C. elegans has a non-functional glycerolase repressor, meaning it never has its glycerolase gene repressed. It's always turned on. It's always activated. It's always making glycerolase, which sounds like a good deal, but when you don't need it, it actually is a waste of energy. You need ATP to run most chemical reactions, and so you're wasting ATP when you don't need to. So I'm going to fill in this chart for you, then I'm going to give you another example and have you fill in a chart. So if we have a wild type C. elegans in a low salt environment, you're going to see that that glycerolase gene is repressed. So there's no catalysis of glucose to glycerol. And again, you then are saving ATP because you're not making stuff you don't need. But in a mutant C. elegans, even in the low salt environment, that glycerolase gene is going to be activated. And so you are going to see a catalysis of glucose to glycerol. Once you put the wild type C. elegans in the high salt environment, though, that stimulus is going to activate the glycerolase gene, and you will get glycerol being made from glucose. And then, of course, you're going to see the same thing in the high salt environment with the mutant because they're always making glycerol. And so if you were making cell diagrams based on this, you already have the wild type. The difference in the mutant is going to be that you're not going to have that dehydrated phase, simply because the glycerolase gene is always activated. Alright, so now let's take a look at an example in humans. So insulin and glucagon are two hormones that are sort of opposite sides of a homeostatic response in humans. Probably in other animals too, but well, I know that other animals have insulin, so they must have glucagon. So we have these two molecules. We know we have glucose, and we get that from digesting our food, and then we know that we store energy in glycogen. So those are those really massive glucose polymers. And the way glucose becomes glycogen is using this hormone insulin, which we've all heard of. And so when your blood sugar, right, that's your blood glucose, the glucose in your blood, increases, your pancreas produces insulin, which helps to polymerize the glucose into glycogen. On the flip side, when your blood sugar or your blood glucose drops, so you don't have enough glucose to sustain everyday function, right? You need that glucose for ATP. Your body releases glucagon. I believe glucagon is made in the liver. And then what the glucagon does is it breaks down the glycogen into glucose so that then you have more glucose in your blood. So that's right, we talk about homeostasis being an optimal range. What we're looking at is an optimal range of glucose. Too much glucose and your body needs to produce insulin. Too little glucose and your body needs to produce glucagon. So there are some forms of type 2 diabetes which are caused by insulin mutations. And what those do, and we don't need to get too into it, but they cause the B cells. B cells are the cells that produce insulin. So when somebody has diabetes, it's often that something is happening to their B cells, so they can't produce insulin. Because of these insulin mutations, the insulin, literally insulin's a protein, it has a different shape, it causes the cell to undergo apoptosis. Apoptosis is when cells blow themselves up because something's wrong with them. So this would be another reason a cell would self-destruct, because there's an insulin mutation, and it's getting stuck in the endoplasmic reticulum as it's being made, and so then the cell blows itself up. Don't worry about that much detail, but the idea being some forms of diabetes are caused by an insulin mutation. Insulin, as I said, is a protein, so it's made by a gene, which then causes B cells to undergo apoptosis. Then you don't have enough insulin. So pause the video and copy down this chart and think about how, and not think about, but fill it out and see if you can figure out what would happen in each case. And then unpause the video and I'll give you the answers. Okay, so if you are not diabetic, meaning you are producing functional insulin and you have a high amount of glucose in your blood, insulin is produced, meaning glucose becomes glycogen. If you are diabetic, meaning, and in this case, this is not all forms of diabetes, but in this form, you're producing non-functional insulin, then you don't have the insulin you need and your glucose stays in your blood. And that's when they talk about like high blood sugar. If you are not diabetic and you have low amounts of glucose in your blood, insulin is not produced so that the glucose stays in your blood. And in fact, if your blood sugar is really low, then glucagon may be produced, and that would increase the amount of glucose in your blood. And then if you're diabetic with this form of diabetes, and you have low amounts of glucose, still non-functional insulin, but in this case, is not produced, right? Now here's the stimulus. Here the insulin, the non-functional insulin, is not going to be produced, so the glucose stays in your blood. So understand that difference between non-functional insulin is produced and the glucose stays in the blood versus the non-functional insulin is not produced, so the glucose stays in your blood. So you can see you can have two effects. Actually, in this case, we have three different, or one effect from three different causes. Okay, in each case, glucose stays in your blood, but in one case, insulin is not produced, in one case, non-functional insulin is not produced, and in one case, non-functional insulin is produced. And this is the problem here with diabetes, right? Because here, it doesn't matter that it wasn't functional because you wanted the glucose in your blood. This is where there's a problem. All right, please ask questions if this isn't making sense, because these notes were really created to help bring everything together in this unit. And so if you're still kind of figuring, I'll figure it out later, you are out of time and you need to figure it out now by asking questions. Have a good night.